begin, I want you to imagine you step outside tonight and look at the sky, and this is what you see. Something's wrong with the stars, right? Stars are not regularly spaced. From our perspective here on Earth, stars are randomly placed. This is just one example of spatial patterning. My name is Connor Healy, and I'm a PhD candidate in Dr. Tara Dean's lab at the University of Utah, and I study spatial patterns, but not in the sky. I study how spatial patterns influence biological systems. And today, I'm going to tell you about a statistic I invented and how I applied it to make discoveries in the bone marrow. The bone marrow is very complex, and spatial patterning plays a large role in its primary function, which is the maintenance and regulation of hematopoietic stem cells. Hematopoietic stem cells are the source of all blood and immune cells. So they can differentiate down both of these lineages. They can proliferate. They can cycle to and from the bloodstream, and they can switch between a quiescent and an active state. Understanding the balance between hematopoietic stem cell fate decisions is critical to human health. If you have too many quiescent hematopoietic stem cells, you get anemia. And if you have too many proliferating stem cells, you can get leukemia. Our hypothesis is that hematopoietic stem cell fate decisions are regulated by the spatial patterning of the bone marrow. It's like a pachinko board, because rather than HSCs making the fate decisions, the patterning of cells and other features in the bone marrow guide HSCs down different paths, much like the pegs in a pachinko machine guide discs down different paths. To test this hypothesis, we first started by seeking the answer to this question. Are there changes in the spatial patterning of the bone marrow that correlate with a shift in hematopoietic stem cell fate decisions? To answer this question, we needed something that shifts HSC behavior. Fortunately, there are age-dependent shifts in hematopoietic stem cell behavior. Specifically, HSCs in newborns exhibit enhanced proliferation, cycling, and blood cell differentiation, while hematopoietic stem cells in adults are more quiescent, less proliferative, and have more balanced differentiation. With samples of marrow from different ages, we can test whether the patterning of the marrow is also age-dependent. To do this, I developed a new statistic to study continuous spatial patterns in biological systems. I call this statistic Rolling Signal-Based Ripley's K, or RSRK for short, and it characterizes signal point patterns in histological specimens. Signal point patterns consist of a point process and a signal. The point process is usually cell coordinates, and the signal is an image or intensity function marking another physiological feature, like the vasculature in the bone marrow. RSRK summarizes the distribution of signal surrounding the points in a point process. It has two terms, E and lambda. E is a term that captures the excess signal within a radius r of a randomly selected point in the point process. To compute this, you scan outward from each point and record the amount of signal you observe as a function of the radius of your scan. Lambda is simply the density of the point signal pattern. And finally, I'd like to point out that RSRK uses a moving frame to capture spatial patterning as a function of location. In a bit more detail, RSRK first sets the frame, then computes the observed K statistic. Next, the points are randomly shuffled, and the K statistic under random distribution is calculated. This process is repeated to generate significance intervals. Then, the frame moves to the next position, and this process is repeated until the full image is analyzed. Finally, once the full image is analyzed, the observed K statistic is normalized to the simulated K statistic to calculate the fold excess signal. RSRK reports the type of spatial patterning at a given scale R and position X, and whether or not it's significant versus random. The color indicates the type of spatial patterning. So red indicates that there is a positive spatial association between the point process and signal. Blue indicates that there is a negative spatial association between the point process and the signal. and white indicates that the point process and signal are independent from each other. The x-axis indicates the position of the type of spatial patterning noted, and the y-axis indicates the scale at which a particular type of spatial patterning is detected. And finally, the x's mark results that are not significant versus random. Having formulated and thoroughly tested RSRK, we obtained marrow samples from newborn and adult mice and used RSRK to determine whether there were age-dependent shifts in the patterning of major regulatory cells and physiological features in the bone marrow. But for the sake of time, and because of their known roles in hematopoietic stem cell regulation, I will focus on our results for analyzing megakaryocyte spatial patterning. Megakaryocytes are a result of hematopoietic stem cell differentiation, and their primary function is the production of platelets. However, they are also known to mechanically restrict the movement of hematopoietic stem cells in the bone marrow, and they secrete anti-proliferative cues. First, we assessed the spatial patterning of megakaryocytes with respect to other megakaryocytes. In this analysis, RSRK identified significant spatial association between megakaryocytes at scales less than 30 microns. This suggests that megakaryocytes maintain a consistent 30-micron gap between each other regardless of age. In other words, megakaryocytes are good citizens and they are social distancing. But in the context of the bone marrow, this gap is interesting because it's just wide enough to permit the passage of HSCs, just like the pegs in the pachinko board. Next, we assess the patterning of megakaryocytes with respect to vasculature. In this analysis, RSRK revealed that in the red marrow, where hematopoietic stem cells are most dense, megakaryocytes are enriched within 20 microns of the vasculature, but only in adults. Increased megakaryocyte density near vasculature could create a physical barrier that reduces hematopoietic stem cell cycling in adults. If there are more megakaryocytes near the vasculature, it's harder for hematopoietic stem cells to get in there. This could explain the observation that hematopoietic stem cell circulation is reduced in adults, and gives us just one more clue how the bone marrow might keep the balance to maintain human health using spatial patterning. In summary, we developed a novel new statistic, rolling signal-based Ripley's K, to study continuous spatial patterns in biology. And using RSRK, we were able to detect changes in the spatial patterning of the bone marrow that predicted changes in hematopoietic stem cell fate decisions. Thank you.